Well, Kisuk Kukits, welcome everyone. My name is Craig Stefani, and I'm a lead with Invasive Species Council of BC. Today, I am joining you from Cranbrook, which is located on the traditional unceded land of the Ktunaxa people, where I'm very thankful to be able to help steward the land and support communities across the province in doing the same. As a provincial organization, ICBC also acknowledges the territories of the Indigenous peoples from across BC, where we live and work to maintain healthy ecosystems for all. So we're winding down May, which is Invasive Species Action Month here in BC. It's an action-packed, dynamic month-long campaign that's used to generate awareness and engage the people living in BC on invasive species. For this year, we wanted to focus our first 4 weeks of ISAM around our behavior change programs, which are used to target people, their hobbies, and their habits. For this final week, we're focusing on Clean Drain Dry, a program to help prevent the spread of aquatic invasive species. Boats and other watercraft, including kayaks and canoes, can easily and frequently carry invasive species from one water body to another. And these aquatic invaders then spread into BC's beautiful lakes, streams, and wetlands. However, boaters, anglers, and paddlers can all take action to help stop the spread by practicing Clean Drain Dry. And today's speaker joins us in helping to explore why it's so important to ensure invasive species aren't tagging along. Please allow me to introduce Brielle Comartin, a passionate master's student at McGill University in the Ricciardi Lab, whose research interests revolve around aquatic ecology and their impact on native mussels in Lake Memphremagog, Quebec. Prior to her master's, she completed an honors Bachelor of Science degree in biodiversity and conservation biology at Western University. In her free time, she enjoys backcountry camping, hiking, birding, and scuba diving. And today she'll be sharing some of her research findings on zebra mussels. Although they're not yet present in BC, they do pose a serious threat to the ecology, economy, and infrastructure if they are introduced. And environmental factors can influence the success of the invasive species, making it essential to help develop predictive metrics to assess this impact. Brielle's research aims to develop these enhanced risk assessment tools to help mitigate the impact and spread of invasive species. It's my pleasure today to welcome Brielle as our speaker.
All right, well, thank you very much, Craig, for that introduction. So I'm really happy to be here today for BC's Invasive Species Action Month. So as Craig mentioned, I'm a master's student at McGill University. I do study zebra mussels, so I may have a little bit more bias towards zebra mussels, but I am here to discuss both zebra and quagga mussels in this presentation. I'll touch on mostly their impacts and prevention. So here I just have a picture that I took underwater of some zebra mussels, and then this is just kind of a fun picture I included of myself in my field gear. So without further ado, I'll just get right into it. So starting with, what are we talking about today? So both the zebra and quagga mussels belong to the family Dreissenidae. So I'll refer to them collectively as Dreissenid mussels or just Dreissenids. The scientific name for quagga mussels is Dreissena bugensis, and for zebra mussels, it's Dreissena polymorpha. These sister species are originally from the Ponto-Caspian region in southeastern Europe, and they were introduced to the North American Laurentian Great Lakes in the 1980s, likely from the discharge of ballast water from transoceanic shipping vessels. So large cargo ships in Europe would fill their ballast up with water, allowing larval, juvenile, and adult Dreissenid mussels to enter. Then they would cross the Atlantic Ocean and dump out their ballast water, releasing these species into the St. Lawrence River. So now Dreissenid mussels are widespread throughout Eastern Canada and the US. Being sister species, quagga and zebra mussels look a lot alike, but they do have some subtle differences that can be used to tell them apart in the field. So for quagga mussels, they will not sit flat on their ventral side. Instead, they'll kind of topple over. And they're much rounder in shape compared to zebra mussels. They tend to have dark concentric rings on their shell and be paler in color at the hinge. However, color is not always the best identifier because the color patterns of zebra mussels varies quite a bit, so it's better to really look at shape. So zebra mussels will sit flat on their ventral side and they are more triangular in shape. Now, comparing Dreissenids to native mussels, they're quite different. For one, native mussels like this BC native winged floater mussel are far larger than Dreissenids. they're either oval or heart-shaped. And a major difference is that they're often buried or partially buried on soft surfaces like sand, so they don't form clumps or attach to vertical surfaces like dreissenids. This is just a nice diagram showing the size comparison between Dreissenid mussels and native mussels. So you can see they're quite different. So what are their impacts? Well, starting off with ecological impacts, Dreissenid mussels are capable of extremely intense filtration activity. One single Dreissenid mussel can filter up to 1 liter of water a day. So by filtering out the phytoplankton from the water column, they're reducing the amount of food available for native fishes and invertebrates. This intense filtration activity also creates clearer water, which allows sunlight to penetrate deeper, promoting the growth of aquatic plants, sometimes invasive plants like Eurasian milfoil, and this also encourages algal growth, which can lead to algal blooms. Also, dreissenids accumulate many pollutants, which can increase pathogenic bacteria, as well as avian botulism through direct consumption of the mussels. And because Dreissenid mussels filter so intensely, they produce a lot of waste, which alters sediment quality, affecting fish spawning and egg survival due to localized anoxia, which means a reduction or absence in oxygen. And then one of the most conspicuous and severe impacts of Dreissenid mussels are on native mussels through biofouling. So that's when dreissenids colonize the exposed surface of native mussel shells, and this can cause suffocation and starvation, ultimately leading to the death of the native mussel. So dreissenid mussels are responsible for severe declines or even complete extirpations of many native mussel populations in the Great Lakes, for example, and in the Upper St. Lawrence, just to name a few. And this is actually what I primarily study for my master's research. Moving on to the socioeconomic impacts of dreissenids, because of their ability to attach onto virtually any solid surface and to do so in dense colonies, dreissenids cause millions of dollars in damage every year in Canada by clogging, for example, intake structures and power stations, water treatment plants, and by damaging watercrafts. So as Craig mentioned, fortunately to date, there are no reported introductions of Dreissenid mussels in BC's lakes or waterways. However, if introduced and established, there would be serious ecological and socioeconomic consequences. Probably the most at risk is the collapse of valuable salmon populations like sockeye salmon. This is because of the ability of Dreissenid mussels to completely alter aquatic food webs, as I explained earlier. And they're also a particular threat to native mussels, especially those that are already endangered, such as the Rocky Mountain ridged mussel, and then also to other native biodiversity. They would also reduce drinking water quality because they encourage the growth of algae, leading to possible algal blooms. They're also a threat to recreation and tourism, which may not seem obvious, but if you've ever been in a body of water where Dreissenid mussels occur and you're not wearing any sort of exposure protection like water shoes, they're extremely sharp and can do some damage to your feet. Probably the most obvious socioeconomic consequence would be the increased maintenance costs of having invasive mussels in your water bodies due to clogged pipes, water intake systems, and municipal water supplies. And so for BC, this is an estimated cost of $43 million per year, and that's not including damage to personal watercrafts or to the commercial fishing industry. So how do they spread? Well, firstly, it's important to mention what makes dreissenids such good invaders in the first place. So their invasion success has been attributed to, firstly, their early maturity. So their lifespan is no more than 4 to 5 years, and females begin to reproduce within 6 to 7 months of settling. And they also have a high fecundity. So a single adult female is capable of producing over 1 million eggs each year. And they have a free-swimming larval stage. So larvae will drift with water currents for several weeks before settling on solid surfaces as adults. And they do so through what is called byssal attachment. So the larvae will metamorphose into adults that will attach onto solid surfaces using adhesive byssal threads. And then lastly, they have a really long survival time out of water. They can actually survive out of water for 3 to 15 days during average summer conditions based on the humidity. So this has major repercussions for overland dispersal. So for example, adults can spread via boats, seaplanes, docks, buoys, vegetation, pretty much anything that they can settle on in the water. And then larvae, being free swimming, can spread via bait buckets, ballast water, dive gear, and even waterfowl. So basically, dreissenids can spread via anything that moves from one body of water to another. And with that being said, recently, my research supervisor, Dr. Anthony Ricciardi, just this past year in Lake Temiscouata in eastern Quebec, discovered the first field documented case of a zebra mussel attached to a fish. So this is a lake chub, which is a common bait fish used to catch other larger, more desirable fish. So what this means then is that the spread of zebra mussels could occur through fish migration. So this fish could carry the mussel upstream. And there's also the possibility of overland transport if fishermen decide to bring this bait fish to another body of water, for example. So now I want to talk about how do environmental factors play a role. So I'm going to introduce you to a kind of science theory called the environmental matching hypothesis. So recently, researchers are becoming aware of the fact that impacts created by invasive species can be context dependent. This is because variations in abiotic conditions, so for example, temperature or water chemistry, can significantly influence the fitness or survival of all species within an ecosystem. So this theoretical framework demonstrates the environmental matching hypothesis, which predicts that an invasive species' impacts within an ecosystem will be the greatest when they are present in environmental conditions matching their environmental optima. So this means that if an invader establishes in an environment where conditions are optimal, their impacts within this ecosystem may be amplified. We're also living in the age of the Anthropocene, where ecosystems are currently experiencing abiotic shifts from human influence and climate change, and they'll continue to experience these abiotic shifts over years to come. So this means that even if an environment right now is not the perfect environment, if environmental conditions change to match the physiological optimum of invasive species in their invaded environments, again, this could lead to an increase in impacts. So for the environmental variables that mediate dreissenid impacts, the most important are substrate size, pH, water depth, and the calcium concentration of the water. For both zebra and quagga mussels, their population densities are highest on large substrates like big boulders or bedrock. And so their impacts will be greatest in environments with large stable substrate. Zebra and quagga mussel populations will have the highest densities in waters with high pH levels, so those greater than 7 or basic pH. And so similarly, their impacts will be greatest in alkaline waters. So water depth is where they differ a bit, where zebra mussel densities will be the highest in shallow waters, and quagga mussels prefer deeper water. So for example, if you look at a breakwall or a dock, you'd likely find zebra mussels kind of at the top where the water just reaches, and then quagga mussels would be underneath the water at deeper depths. Probably the most influential environmental variable is calcium concentration. And this is because dreissenid mussels require calcium for proper shell growth and production, as well as osmoregulation. So zebra and quagga mussel population densities will increase with increasing calcium concentration of the water. So impacts will be greatest where calcium concentrations are the highest. So this is also a really large part of the research that I do for my master's. I wanted to show you really quickly this table that demonstrates the description of the levels of risk establishment of the zebra mussel in a lake based on its calcium concentration. So this is based on Quebec lakes, but it applies to all water bodies. So you can see in green that below a calcium concentration of 8 milligrams per liter, there is absolutely no risk of zebra mussel establishment. And this is because adults cannot survive in this low of calcium. They need at least 8 to 12, and even then, the survival of zebra mussels is low, and they certainly cannot reproduce. The optimum calcium levels for all life cycle stages of zebra mussels is 25 milligrams per liter and above. So if a water body has this calcium concentration, then there's a very high risk of establishment in that lake or river. And so researchers like myself are trying to better understand and predict the impacts of invasive Dreissenid mussels based on environmental variables to mitigate the establishment and potential impacts prior to their invasion. So now I think the question that we're all asking, well, how do we stop them? So unfortunately, once zebra mussels have established, it's already too late. That's why preventative measures are absolutely key. I do want to mention, though, that it is possible to undergo some zebra mussel eradication methods once they have established through manual cleaning and removal. And this is actually taking place in a lake in southern Quebec called Lake Massawippi. They're kind of doing an experiment to see how this works. So they're about 2 years into this experiment. But this can be impossible for large, heavily infested bodies of water. It does use a lot of government resources, and it may not be sustainable because larvae exist microscopically in the water column, so they'll always come back. So it'll be interesting to see what happens with that experiment going on at that lake in Quebec. So this is what Craig was talking about earlier. So this is kind of a Canada-wide procedure to prevent the spread of aquatic invasive species. So the clean, drain, dry. So the easiest way is to follow these 3 steps. So for clean, inspect and clean plants, animals, and mud from watercrafts, trailers, and gear. Drain, so drain all water from your watercraft, trailer, and gear. And then dry, similarly dry all parts of your watercraft, trailer, and gear completely between trips. And this doesn't just apply to boats. This applies whenever you go in, on, or near water, whether you're fishing, boating, kayaking, canoeing, paddleboarding, using waders, or whatever it may be. Different provinces and territories in Canada have varying regulations and requirements when it comes to cleaning, draining, and drying to stop aquatic invasive species. So in BC, there is the Invasive Mussel Defense Program, which was created in 2015, and it has 3 main components. So the first is a watercraft inspection program to detect and respond to high-risk watercrafts, potentially transporting invasive mussels into BC from infested lakes or rivers. And then secondly, they have some lake monitoring programs to confirm the continued absence of invasive mussels in BC waters. And then lastly, they do some outreach and education to educate people and change behavior by promoting the message of clean, drain, dry, which is what we're doing here today. So I just wanted to finish on this last note that there's a lot of information becoming available on the proper disposal of mossballs. So this is the most recent thing that we have to watch out for. Zebra mussels have been detected in mossball products for aquariums. So you might have heard of Marimo mossballs. They're these cool species of green algae popular in the aquarium trade. So the government is now aware of this, and mossball products have been banned commercially in Canada. And they're now encouraging the proper treatment and disposal, which is basically to put your mossball in a freezer bag and freeze it for 24 hours, and then dispose of it in your household garbage. And then to follow up by treating your aquarium contents and water to ensure that there are no zebra mussel larvae. So that's all that I have for you today. So I'll now answer any questions if you have any. Thank you.
Thank you, Brielle, for the amazing presentation. Like you said, we have a few minutes for questions now. If anyone has any questions, feel free to type them into the Q&A box. Just to get things started, you mentioned a bit about your research, and I was just wondering, what does a day of data collection look like for you when you're in the field?
Yeah, so I'm usually dressed like this image here. And so either on a boat or by shore, I'll toss 5 quadrats that are about 1 meter by 1 meter square. And then I'll just kind of toss them haphazardly. I'll go down and I'll dive, and I'll collect every single native mussel within those squares. Then the second part is to process these mussels, which involves removing every single zebra mussel attached and counting them. So this gives me a number of the average number of zebra mussels attached to unionid mussels. And then I can do this at several sites within a lake, which usually is a several-day process, not just 1 day. And then I can compare from year to year to kind of see how the population of unionid mussels are doing. That's what a typical summer day looks like for me.
Winter's a little different?
Yeah, winter is mostly looking at my data, and kind of doing the more not so fun office work. Yeah.
When you are diving, have you seen something that's been like really surprising, or what's like the weirdest thing that you've seen them attached to? Have you come across anything?
Yeah, actually, last year, I forgot what exactly it was. It was some sort of piece of plastic. So just like a broken piece of plastic off of whatever it would have been. And it was just littered in zebra mussels. So it kind of just goes to show that they will literally attach to anything.
Have there been innovations into looking at some of the technologies maybe that look into preventing zebra mussels using their abyssal threads? Because that seems to be one of the big differences between native mussels, is that native mussels don't have those abyssal threads, so they're not attached to things.
Right. No, that's just they just need that for survival. So actually, this is really common with a lot of native marine mussels, like blue mussels. So they have abyssal threads as well, that they'll use to attach. So it's like similar to barnacles, where they need to attach onto something in order to survive. If you cut off their abyssal threads, it's as adults, it's likely that they just wouldn't survive.
With you could mention kind of that calcium concentration is really key to their survival.
Yes.
Do you have numbers for either in your research area or BC or Canada in general, anything like that, for what like percentage of those lakes can actually support them? Is it pretty common to have calcium concentrations at those levels?
Yeah, it is. So where a lot of native mussels have been extirpated, so that's the majority of the Great Lakes and the Upper St. Lawrence River. They have concentrations of calcium above 25 milligrams per liter. So it's very common. The lake that I'm working in, Lake Memphremagog, actually has what would be classified as suboptimal calcium. So it's about 16 milligrams per liter. So that means that zebra mussels can occur there, but they may not be able to reproduce in large numbers. However, a large part of my research is figuring out, well, they have low calcium, yet they're there in large numbers and they are impacting native biodiversity. So we don't really know why. But it's interesting. We might have to kind of shift that risk assessment if I do find some interesting results.
That kind of actually feeds into another question I was kind of wondering about is, in your opinion, then, what are kind of some key research priorities you think in the field of zebra mussels that are kind of like the next step for their management?
Yeah, that's a great question. I think that risk assessments are major. I think that if we can predict before an invasion if they will be successful in that body of water, then that's extremely useful information, because that's kind of where we went wrong with Lake Memphremagog. There wasn't too many preventative measures taking place in terms of boats entering the water because we thought, well, it has low calcium, so we don't really have to worry about it. They're not really going to establish here. So I think if we have a better understanding of the environmental factors contributing to zebra mussel distributions, then we'll be able to kind of put in place those preventative measures before it's too late.
Yeah, it can really help with helping to have the resources in the right places rather than having them spread out further in places that might not be optimal for needing their protection.
Exactly.
With that, then, kind of to wrap up, just on a high note, then, what do you find most rewarding about your research, and how does that inspire you to kind of continue what you're doing?
Yeah, I think it's really rewarding that this is something that a lot of people, especially in Eastern Canada, are extremely familiar with because we see it everywhere. So I think it's rewarding that my research may be able to actually impact policy. Because I think as a conservation biologist, that's kind of the main goal. I want this to go to policy so that we can actually make those changes and prevent further spread, for example, to the western side of Canada. So yeah, being a conservation biologist can sometimes be a little bit discouraging when you're constantly seeing a lot of native biodiversity loss. But at the same time, it's nice to know that my research results could contribute to possibly stopping the spread or furthering this loss of biodiversity.
Thank you so much for joining us today. I definitely have a bias towards them as well. I got my invasive species as part of the invasive mussel defense program. So I'm for that. So thank you for joining, and thank everyone else that's able to join us today for the webinar. Hope you enjoyed yourself and learned a bit more how you can make a difference in your community by practicing clean, drain, dry. Be sure to check out our ISAM page for more details on the month. We are winding down here, but you can look to see if there's any other events coming around BC in your area. This is a link to our store where you can find some sweet bucket hats to help protect you from the sun while you're out on the water. And today does mark the last of our ISAM Wednesday Lunch and Learn webinars. However, you can also check them out if you've missed any on our YouTube page. And I hope you enjoy the rest of your day. And again, thank you for joining us.